Okay, so today we're going to be reviewing the biochemistry and cells portion for the AP Bio exam. So we're going to start a little bit first with the biochemistry and then move into some cells information and then do some practice problems again like we did last time, okay? All right, so just to review, there are 25 different elements that make up, that are considered elements of life, and they're listed here on this table. The main ones to make sure that you're familiar with are the carbon, oxygen, hydrogen, and nitrogen, which are going to make up 96% of life. And then the phosphorus, sulfur, calcium, potassium, and a couple trace elements like iron and iodine are going to make up about 4% of the rest of the life elements. But again, the carbon, oxygen, hydrogen, and nitrogen are the four main ones, but we also like to include the phosphorus and the sulfur as well as part of the 6 main elements to remember as elements of life. So a hint for that is to remember CHNOPS. So that's the abbreviation for those different elements. So carbon, hydrogen, oxygen, nitrogen, oxygen, phosphorus, and sulfur. Okay? Then we're going to review here the atomic structure. So this is very basic. Remember that the atom is the smallest unit of matter, and that's going to retain all the properties of an element. Okay? And within the atom, the subatomic particles you need to be familiar with are the neutron, which of course is inside the nucleus and is considered neutral as far as the charge is concerned. Neutron has the word neutral in it, so neutral makes sense. Its mass is approximately 1 AMU. Protons are also inside the nucleus and have a positive charge, and their mass is also 1 AMU. And then electrons have a mass that's very too small really to detect, so we don't usually consider it as having mass, although it has to be in existence, but it's very negligible. It is found outside of the nucleus in the electron shells, and its charge is negative. When we talk about bonds between those electrons, because that's where the bonds take place, we have 3 main ones that we're going to discuss here. We have the covalent bond, the ionic bond, and the hydrogen bond. And all of these are very important to life and how things interact with one another. Covalent bonds are going to be found forming in between the cell's main molecules. So when we talk about things like carbohydrates and lipids and proteins, covalent bonds tend to be a primary bonder in those major complex compounds. They're very strong bonds, whereas the ionic bonds are going to be a little bit weaker, and they are going to allow for quick reactions and responses. Because of them being weaker, they can be broken or put together pretty easily. Hydrogen bonds are the weakest out of the 3. And they are only found between hydrogen atoms and other electronegative atoms. Okay, so mainly oxygen, but other electronegative atoms as well can have a hydrogen bond. And all bonds are made and broken through chemical reactions. There is a 4th type of bond that is an even weaker bond that is mentioned, so I just wanted to bring it up, and it's the van der Waals interactions. These are very slight and fleeting attractions between atoms and molecules that are close together. It is the weakest bond out of all of them. The example here is if this is a gecko, and the gecko's toe hairs with the wall surface is considered a van der Waals interaction or bond. And how they are able to kind of stick to the wall and kind of climb on it. When we talk about the hydrogen bonding, it's very important to start to talk about water and how the hydrogen bonds and the polarity that happens with their bonds plays an important role on some of the different characteristics of water. So when water is formed, the hydrogen and oxygens, there is not an equal pull on those atoms. So the oxygen tends to have a negative charge, whereas the hydrogen tends to have a positive charge. And when the hydrogen combines to the oxygen of another water molecule, that is considered a hydrogen bond, as well as when the oxygen of one attaches to a hydrogen of another. So any connection between one water molecule to another is hydrogen bonding. And that is due to the polarity that is going to happen in the original molecule. So water can actually form up to 4 additional bonds with itself. And this is going to lead to those unique water properties that are very important to life: cohesion, adhesion, surface tension, specific heat, evaporation, and universal solvent. So cohesion and adhesion are going to be the stickiness of the water molecule due to that polarity. Cohesion is the like with the like, so that's basically going to be where the water is connected to itself, okay? And that's due to that polarity and hydrogen bond. And this is what helps water kind of go up against gravity in plants in the vessels so that they can go from the roots up to the tall trees. Also, adhesion is another stickiness. This is between unlike, so this is water with something else. And this is evident in our blood, how our blood is able to kind of stick with the veins as well as plants as well. You get a little bit of that as well. So the cohesion and adhesion takes place in both of these, or both take place in plants. I'm sorry, that's what I meant to say. Surface tension is where we get that little bit of like skin on the top of the water, which is how bugs are able to walk on water, and that's what forms drops in raindrops. And when you spill water, it tends to form like a little bubble, and that's also due to the hydrogen bonding between the water molecules. Specific heat is what allows water to absorb and retain energy so that it doesn't change temperature very quickly. So it helps keep our ocean at a moderate temperature. It helps protect our temperatures of our marine life that are living under ice, as well as in the oceans and near the equator. Evaporation is another property of water, where the liquid evaporates into the gas, which helps with the cooling mechanisms of organisms and helps us maintain homeostasis. And then, of course, because of the polarity of the molecule, it is able to attach to other substances and ions very easily and break them apart. So that is why it's considered the universal solvent, because it's able to dissolve and break apart other molecules very easily. So those are some main important properties of water. Again, solvent, it means that something that can dissolve something else, and the rule of thumb is that like dissolves like. So that means hydrophilic substances will dissolve hydrophilic substances, whereas hydrophobic will dissolve hydrophobic. So some characteristics of the two different types of substances: hydrophilic is going to have an affinity for water. This is something that will be able to attract water, so to speak, and dissolve well in water. So it's usually polar and involves ions. Things that are examples that would be hydrophilic are, of course, water itself, cellulose, sugar, salt, blood, that type of thing. Hydrophobic, phobia means fear, so this is water fearing, and so it appears to repel the water. So it's considered nonpolar, and this is things like your oils and your lipids, and this is found in the cell membrane. Your cell membrane is that bilayer of the phospholipids, so it's primarily a lipid composition. Moving on to acids and bases, remember that acids are going to release hydrogen ions into solution, so they add hydrogen ions, and their pH on the pH scale is less than 7. Bases, on the other hand, are going to release OH negative ions or hydroxyl ions, and their pH will be greater than 7. And then a buffer is something that can minimize those changes in the acids or bases in a solution. And basically they're usually weak acids or weak bases. So anything close to that 7 on the pH scale is usually considered a buffer, because it can help stabilize and neutralize the change of a solution from an acid or a base, a strong acid or base. So they help keep our blood at a pH level of 7.4, so our blood is slightly basic. One example of a good buffer is a bicarbonate, which is something that our pancreas does secrete. So here's the pH scale with just some examples of some aqueous solutions that are at the different levels. You can see most of your cleaners are bases, and we've got our citric acids are up here in the acid level. So tomatoes and lemons, all of those tend to be acidic. Oranges. Our urine is actually slightly acidic, which is one of the things that helps keep it from being helps to keep it sterile. Most people don't realize that urine is sterile unless, of course, you have some sort of a urinary tract infection, but it is a good source of a sterile solution if necessary in a tight spot. Pure water, of course, is neutral, but that's hard to find. Most water is going to be slightly acidic or basic depending on the components also found in it. And then, of course, human blood is on here as being slightly basic because it's at 7.4. All right, so now we're moving into basically the formation of our large compounds and molecules. So we got to remember that monomers are your small organic building blocks. They're single small building blocks. I guess I'm trying to think of another way to put that wording, but that's the only way I can think of. And they are going to come together to form our larger polymers, which are the larger molecules. So how they come together is usually through dehydration synthesis, which we're going to talk a little bit more about in the next slide. And they're usually connected with a covalent bond. So the polymers are going to have many of the monomers together, and usually those monomers are identical or at least very similar. And then when we put multiple polymers together, we can get a macromolecule, which is what we would consider something like a protein or carbohydrate. So here's an example. You got the amino acid would be considered the monomer of a protein because it's the smallest building block. Many amino acids put together form peptides, which are longer strands of amino acids. And then if you have a long peptide or lots of peptides put together, we get the polypeptide. And then the polypeptides come together to form a protein. So that is the protein would be the macromolecule. So ultimately it's from very small to much larger. Pretty common sense there. So hydrolysis and dehydration synthesis are just the main overall terms given to the forming or breaking of these large polymers and macromolecules. When we make a polymer, which means we're putting together the monomers, we're adding them together to form a larger molecule. The water molecule is going to be released in that process. So when the A and the B come together, water is released as they become that larger polymer, and so that's why it's called a dehydration synthesis. Synthesis means to come together. Dehydrate means to pull out, so the water is being pulled out, and so that is why water is released, and that's where that name comes from. When we break down a polymer, so if we were to break down a peptide into the smaller amino acids, then we're going to have to put water back in because that oxygen and hydrogen has to be put back to those simple monomers individually. And so that's why that's called hydrolysis. Hydro means water, lysis means to break apart. So water's going in to break apart. Any questions so far? Okay. All right. So now let's talk about those four main macromolecules that are very important to life. So the first one is carbohydrates. Remember that these are used for building. So they are used for structure. For example, cellulose in the plant would be an example of that. And they are also used for fuel. So the sugar is the main chemical that is going to be converted to ATP for the cells to be able to do work. They are building blocks. Their building blocks or monomers are the monosaccharides. So that's a single sugar. And then those combine to form disaccharides, which would be a double sugar. And then from there we would get polysaccharides, which are many sugars put together. And then the polysaccharides end up becoming our macromolecule carbohydrates. Okay? So glucose is actually considered a monosaccharide. It's a very simple sugar just by itself. An example of a disaccharide would be fructose, sucrose, that type of thing. And then our polysaccharides here are starch, glycogen, cellulose, and chitin. So for storage, plants use starch as the polysaccharide way of storing, and animals will store carbohydrates as glycogen. And for structure, plants use cellulose that's found in the cell walls of their plant cells, whereas in animals, we tend to find our carbohydrates used as structure in exoskeletons in the form of chitin. And basically, it's just a difference in the arrangement or the orientation of these monosaccharides and the position that they're in those polysaccharide combinations that determines which of the molecules it becomes. It has to do with its arrangement and sequence. Lipids are, of course, your fats primarily, and they're used to store large amounts of energy as well as to help with insulation and protection. We can have saturated fats, which means that in the fatty acid chains, all of the bonds are surrounded with hydrogen, so there's no openings. Unsaturated means that there's going to be an opening, and there's going to be some double bonds, basically, in the fatty acid chain to allow for some openings around the chain and have some holes where hydrogens are not going to be found. And then polyunsaturated would mean there's multiple holes, so multiple double bonds. Steroids are another type of lipid, and so examples of those are cholesterol and hormones. Those are different types of natural steroids that we have in our body. And then phospholipids, of course, are a very important lipid because they make up the cell membrane primarily. Not the only thing in the cell membrane, but they're a big part of it. And you can see down here the picture of the single phospholipid. It's got that hydrophilic head, which is water-loving, and that's going to be the circular portion, mostly phosphates. And then it's got the hydrophobic tail, which is where all the lipid is, which is water-fearing. And it's a bilayer, so we have two layers of these little, they look like hairpins almost, these phospholipids, and that's what keeps the cell separated from the outside environment. Now when we talk about proteins, proteins have so many different functions in the body, so we have to kind of talk about their structure, because the structure is what determines the function of the protein. So proteins have 4 different levels of structure. The primary level is just the amino acid sequence. So there's 20 different amino acids in nature, and the way that those amino acids come together, the number of amino acids that you have and the different sequences are what determine what kind of protein you're going to get. So that's why we can have so many different proteins even only out of 20 amino acids. And the bonds between those amino acids are called peptide bonds. And so you can see this is this picture. Let me actually get something to write with here. What do I want to use? All right, so this one right here is going to be your, oops, no, I don't want to draw another circle. Really. Sorry. Trying to, there we go. All right, so that is basically our primary. And it's just a chain of amino acids. It's a little small, but I have some bigger pictures on the next slide. Our secondary level of protein structure is where it starts to gain a 3D shape, so it starts to fold and coil by hydrogen bonding. So the hydrogens are going to come together. This is where we get an alpha helix or a pleated sheet. So this is actually shown up here in this picture. Okay, so we have the helix showing right here, the alpha helix, and then over on this side is the pleat, which it looks kind of like a accordion. So that's where we start to get our, I'm not very good with writing with this pen on here. So that's where we start to get our 3D shape to start forming. And that's because the hydrogen bonding that's going to start to occur between the different parts of the protein. And then we get the tertiary. So at the tertiary, we start to get bonding between the R groups of the actual amino acids. And so those bonds are either going to be hydrogen bonds, or we also will get some disulfide bridges, which are just basically bonds between two sulfur atoms. So that's shown here. And again, I have bigger pictures of this on the next slide so you can actually see those bonds that I'm talking about. So that's the tertiary level. And then the quaternary level is going to be where two or more polypeptides actually come together, which is shown in these images right here. Okay, so that's where we get the final shaping of the actual protein. And based on what that is going to determine what the function of the protein is going to do. And proteins have tons of different functions within our body. So here's a bigger picture here showing you how we go from amino acids to polypeptides to protein based on the shape. We start off with the primary structure with just the sequence of amino acids. Then it gets folded into a 3D structure with that secondary structure either being a helix or the pleated plate, the pleated sheet, which is going to just be because of hydrogen bondings happening between hydrogen and oxygen going on between the different strands of amino acids. And then we start to get folding in on itself again with the tertiary structure where the actual bonds between the R groups of the different amino acids will start to bond. So you'll get a lot of sulfide bonds as well as some more hydrogen bondings. And then finally that quaternary structure, which is the final overall structure that determines what it's going to do. Okay? All right. Again, protein structure and function are very sensitive and to their chemical and physical conditions, so it's important to remember that they have to have very specific pH and temperatures to maintain their function and work properly. If the pH or the temperature is not optimal for that particular protein, then it will denature, which means that it basically becomes non-functional. It will actually unfold, which you see happening in this picture here. And when it starts to unfold and lose its shape, then it can no longer function properly, so it's no longer able to do its job. So that is why temperature and pH are very important in maintaining the function of proteins. When you go to when you go to cook an egg on the pan, so you have your raw egg and you put it on the hot pan, that temperature becomes too high for the proteins in the egg, and that's why you start to see the actual egg whites harden up and the yolks start to harden up. That is actually from the denaturing of the proteins that are in the egg. So that's just an example of how that works. We like it because we like to eat it that way, but it means that the egg won't be able to do what it needs. If it were to actually become a living thing, it would no longer be able to do so. All right, looking at nucleic acids. Nucleic acids store all of our genetic information, and the monomer of a nucleic acid is the nucleotide. So it's important to remember that there are two main nucleic acids, DNA and RNA. DNA is a double helix, RNA is a single strand. When we talk about the bases that are found in those different molecules, and we're looking at the nucleotides, DNA has thymine, whereas RNA does not have thymine. It has uracil instead, but otherwise they both do still have the cytosine, guanine, and adenine. DNA is going to, of course, carry the genetic code, whereas RNA is more involved in being the messenger of that code and helping to turn that into the protein. DNA is much longer and larger and does have the sugar deoxyribose, which is where the D comes from in DNA, whereas RNA, it's an R because it is ribose as the sugar and not deoxyribose. And its main function is, again, to take that message that the DNA has and translate it so that a protein can be made. All right, so now moving on. Any questions about those four major macromolecules? Okay. All right, so moving on, we're going to talk about light microscopes versus electron microscopes, just as a quick overview. Just remember that when we talk about light microscopes, these are the microscopes that we can actually see a living or use a living specimen, because it's just light that is being used to view the specimen. However, because of that, the magnification is very limited to only about 1,000 times, okay? Whereas electron microscopes give us much greater magnification, up to 1,000,000 times. But to do that, the specimen has to be in a vacuum, which means that it's going to have to be non-living, because the vacuum would kill it regardless. And it uses electron beams through the specimen to give the image, which is generally black and white. All right, now we're going to move into the cells. Now that we've talked about the microscope, which is what allowed us to discover cells, there are two different types of cells based on whether they typically have a nucleus or not. So we have prokaryote versus eukaryote. You carry the nucleus. Eukaryotes are the ones that have an actual nucleus with a nuclear membrane, whereas the prokaryotes are our primitive cells that do not have a defined nucleus. They do still have DNA in the cell, but it's going to be in what's considered a nucleoid. It's basically just a circular strand of DNA with maybe possible small circular pieces of DNA spread out throughout the cell in these small little capsules. They both have cytosol or cytoplasm. Excuse me. But the prokaryote's not going to have any membrane-bound organelles. They will still have ribosomes like a eukaryote, but as far as endoplasmic reticulum, Golgi apparatus, any of those other membrane-bound organelles will not be found in a prokaryote. Those are only found in eukaryotes. Prokaryotes, of course, then are much smaller and are bacteria. Eukaryotes are going to be much larger and more complex and are found in plants and animals as well as protists. So that's the main difference. So again, prokaryotes, no defined nucleus and no membrane-bound organelles besides the ribosome, but they do still have cytosol and they still have DNA. So here's a generic picture of an animal cell, just kind of showing you all the different possible organelles that could be found in an animal cell. So you have the nucleus here in the center, surrounded by the endoplasmic reticulum, and then we of course have a Golgi apparatus. You've got some mitochondria in here. We've got the cytoskeleton, which helps maintain shape. We've got lysosomes, the plasma membrane. Inside the nucleus you have a nucleolus. You have the chromosomes or the chromatin. Some plant cells might have a flagella, which is a singular little tail, kind of used to either move the cell through wherever it's going. That's the main function of a flagellum, is for movement. Microvilli, some cells have microvilli, which are these finger-like projections that help increase surface area for absorption. They also might have cilia, which are even smaller projections that help with usually movement of dust and debris alongside the outside of the cell. Peroxisomes are kind of similar to lysosomes. The centrioles or centrosomes help with cell division. So these are all the possible organelles that can be found in a cell, in an animal cell. It's important to remember, though, that not every animal cell will have all of these, and some animal cells have more of one organelle than another depending on what it does. Like, for instance, a muscle cell is going to have a lot more mitochondria than maybe a cell that's in your skin, and that's because your muscles are going to have to exert and use more energy, so those cells need to be able to do that with multiple mitochondria. Nucleus, they all will have a nucleus at least for a portion of their lifetime. It's important to remember that red blood cells do not have a nucleus for their entire lifespan. They will have a nucleus at the beginning, but then eventually eject it out so that it can make more room to hold on to oxygen, to have hemoglobin hold on to oxygen. But that also is one of the reasons why a red blood cell has a very short lifespan, because once that nucleus is gone, it doesn't live much longer after that. Let's see, what else? So those, just some things to kind of keep in mind. Obviously, the only human cell that has a flagella is the sperm cell, so no other animal cells will have a flagellum. Microvilli are primarily found in cells in the digestive system. Cilia are mostly only found on cells in the respiratory system. So you can see it, depending on the function of the cell, will make a huge impact on the different organelles present. Okay? Whereas in the plant cell, same type of thing. Plant cells, depending on where the cell is in the plant, will depend on the amount and types of organelles. But again, here's some of the things that a plant cell will have. You can see that there are a lot of similarities. Plant cells also will have, of course, that nucleus, because it is a eukaryote. It will also have a nucleolus and the chromosomes, rough or smooth ER, the endoplasmic reticulum, Golgi apparatus can be found. We'll also have ribosomes, which I don't think I've mentioned in the animal cell, but animal cells have ribosomes as well. Mitochondria are going to be in animal cells and plant cells, so you can see those here. Peroxisomes are also here in a plant cell. Some things that plant cells do not have are lysosomes. So plant cells do not have lysosomes. They do not have centrioles. Okay, they do not have centrioles. And usually they do not have flagella. There occasionally might be a plant cell that has a sperm cell that might have flagella, but majority of plant cells do not have the flagella either. Okay, but the two main ones to remember that plant cells do not have is the lysosome and the centrioles. Now, they also have some things that animal cells don't, and that is the chloroplast. So animal cells will not have these chloroplasts because they do not have to harness the sun for photosynthesis. Plant cells do not have a cell, animal cells will not have a cell wall, which you can see is found in the plant cell here. Okay, cell walls are only in plant cells. The large central vacuole is another thing that is unique to a plant cell. Animal cells will have some small vacuoles, but the large central vacuole is only found in the plant cell. They both have cytoskeleton. I think that covers, they both have a plasma membrane. So a lot of similarities, but there are a few differences between the two. When we talk about plant and animal cells and their size, it's important to remember that they can only get as large as they can handle the waste and materials coming in and out of the cell. So they have to maintain a relatively small shape because they need to make sure that the surface area is larger to the volume of the cell as far as a ratio is concerned, because if the volume of the cell gets to be too large, then the surface area of the cell cannot keep up with the exchange of the materials between itself and the environment. So the waste will build up, and there won't be enough materials to be able to be brought in for the cell to be able to function properly. So this is just a chart kind of showing you how that works. As you can look at these cubes, as we increase the size of the cube, the surface area will increase, but the volume increases much faster, so that the ratio becomes smaller. So at the 2 centimeter cube, we have a much larger surface area to volume ratio, a 3:1, which is what we would want. The next one up, the 4 centimeter cube, we can see that that ratio has decreased. Okay, it's only 1.5 to 1. So it still has a larger surface area, so it's still probably be functionable, but we're getting tight. Then we get to the 6 centimeter cube, and it's a 1-to-1 ratio, so this is where we'll start running into problems. And then that 8 centimeter cube, the volume has now exceeded the surface area, so the ratio is 0.75 to 1. So as the cell increases and gets bigger, that surface area to volume ratio decreases. So it's going to be harder for it to be able to maintain that the fusion and exchange of materials with its environment. Okay. Any questions so far? Okay. All right, so this is just the next couple slides just kind of review the function of some of the major organelles in plant and animal cells. So we got the nucleus here. It's just important to remember that it is surrounded by a nuclear envelope, which is going to control and kind of deal with the movement of stuff in and out of the nucleus and make sure that the chromosomes, the chromatin within the nucleus does not come out. And it is going to connect directly to the endoplasmic reticulum of the cell. So they are connected. The nuclear membrane is connected to the ER. The nucleus' main function is just to house that DNA and the chromosome material, and also to house the nucleoli, which is the smaller circular part here that's a little darker, and that's where ribosomes get made, and those exit through the little pores that you can see here on that membrane nuclear envelope. Ribosomes are basically RNA. Primarily, that's what they're made up for, and they are going to be involved in the protein synthesis, the making of the proteins and actually building of the proteins. So that's their main function. The ER, the endoplasmic reticulum, there is smooth and rough. There's two different kinds. The smooth, of course, does not have ribosomes attached to it, and that's why it gets its name smooth. The rough ER does have ribosomes attached to it. So that's the difference in the names. As far as functions, the smooth tends to play a bigger role in helping to synthesize lipids as well as metabolize carbohydrates, whereas the rough ER is going to play a very large role in the synthesis of proteins as well as the movement of those proteins. Okay. Golgi apparatus is going to be this little series of flattened sacs with little vesicles that kind of pops off of them, and that's because it is primarily involved in the modification of the proteins and carbohydrates as well as the sorting of those products and the movement of those products. It helps to release products out of the cell when necessary by transporting through the vesicles. So it's kind of like a package warehouse there. Lysosomes are small little sacked membrane organelles that are filled with hydrolytic enzymes. Lyse means to split apart, so a lysosome is involved in the breakdown of substances. Usually plays a role in recycling of damaged organelles. So any of the organelles that have become damaged or overused and need to be broken down in the cell, the lysosome will take care of that, as well as any macromolecules that need to be broken down as well in the cell. Vacuoles are going to be found primarily, like I said, the large vacuoles in the plant cell. There are small vacuoles that will be found in animal cells. They help with storage primarily, and in the plant cell, it's a huge role in the water balance and the storage of water. Mitochondria, of course, is going to have its role in cellular respiration. It is a double membrane organelle. It kind of looks like a pill bug or roly-poly, but however you know them as. But that's its main job, is cellular respiration. And both plant and animal cells have this because they both have to be able to harness that energy from the glucose and convert it or use that to convert ATP. Chloroplasts are only in the plant cells. Those are responsible for photosynthesis. They actually have, again, two membranes and then the little stacks of grana, the thylakoid stacks in the grana inside. And that's going to be where the different parts of photosynthesis take place. And then peroxisomes are kind of like lysosomes, except that they deal primarily with hydrogen peroxide. So it's just a stack that has a job in helping to kind of break down and convert hydrogen peroxide into water. And then here's just a breakdown of the cytoskeleton. So the cytoskeleton is the tubes and filaments within the cell's cytoplasm that helps to maintain the shape of the cell. So there are three main types of filaments. There's the microtubules, which are tubes. They're actually tubes that are a little thicker. And then you have the microfilaments, which are going to be thin proteins, typically actin. And then the intermediate filaments, which are going to be a little bit thicker than the microfilaments, but they're not quite as big as the tubes. And they all function together to maintain the shape. Some of them also help with the formation of the cilia and the flagella. So for instance, the microtubules are going to be involved in the flagella and the cilia. They help with the movement of the organelles. Let's see. Microfilaments play a big role in muscle contraction. So within our muscle cells, we actually have a lot of actin filaments as well as myosin filaments that help with the contraction of the muscle cell and ultimately the muscle as a whole. And they also play a role in cell division. The filaments tend to have more of a role in that cell division. All right, and then the intermediate filaments, again, they all maintain the shape, but the intermediate filaments also help with the formation of the nuclear lamina and kind of anchoring the nucleus and other organelles in the cell. Now, between the plasma membranes, or the cell membranes, however you want to call them, there are going to be junctions between two or more cells. So these are just some examples of some different types of junctions that we can find in animal cells. A tight junction, which you can see here, is going to prevent any leakage from one cell to the next. Okay, so that's why it's called a tight junction, because it's going to keep leakage from happening. The desmosomes, which are going to be found, let's see here, right here, anchor the cells together. So desmosomes help to anchor them, and that's because they have these little filaments that kind of branch off into each of the cells, so it acts like an anchor to hold the cells together. And then lastly, we have the gap junction, which is here, and that's going to allow for passages of materials through the different cells. Okay, so those are three different types of junctions. And then if we look, here's an overall microscopic view of the cell membrane, or also known as the plasma membrane, because it is a movable creation. It is not rigid. And you can see that bilayer of the phospholipids right here. So you've got the tails face inwards towards each other, forming the middle of the membrane, whereas the phospholipid heads are going to face either inside the cell or outside of the cell. And it's important that we remember that it's often referred to as being fluid mosaic. Fluid because it does move, and that's because of the cholesterol that's found embedded within the cell and the fact that it is made up of a lipid bilayer. And mosaic because there are other things embedded in it as well, such as these proteins. We've got the integral protein, which goes all the way through. You have a peripheral protein, which is going to be just on one side or the other. We also have these glycolipids, which are little like markers that we see on the extending out of the different proteins that help with recognition of the cell to other cells. So it's got lots of different things within it. Those proteins are involved in the movement of substances in and out of the cell. They also, of course, with those glycolipids and glycoprotein or proteins attached to them, are going to help with recognition and to other cells as well, you know, and then in hormones and things like that. So the plasma membrane is involved in the movement of substances in and out of the cell, and so we have two main types of movements. We have passive versus active. Remember that passive means that little or no energy is used, and so it tends to follow the normal flow of concentration movement from a high concentration to a low concentration. This is very, you know, common sense. If you're in a large group, really crowded, the instinct is to kind of spread out and try to get your own space. So that's the same with molecules as well. They tend to want to dissipate away from one another to kind of reach an equilibrium. So that's considered moving down the concentration gradient because of that movement from high to low. Examples are things like diffusion, osmosis, and facilitated diffusion. Active, on the other hand, requires ATP energy to make it happen, and that's primarily because the movements tend to go against the concentration gradient, meaning that it's going to be movements of low concentration into a high concentration area. So it's not the normal. It's kind of like if you had, a large group that was, packed full, and you were still trying to shove people in. That would not be a normal movement, so that's considered active. And it would probably require a lot of energy to do that. So these are your pumps, which involve usually proteins that pump ions and things through. It also is going to involve your Golgi apparatus and the use of vesicles in endo and exocytosis, the movement of large amounts or large substances. I'm going to skip through to this one real quick. We'll come back to that other one. So here's a picture of diffusion, where you can see you have some molecules that just move through that plasma membrane without any help from anything else. You've got the high concentration. There's a lot more little dots up here, fewer inside, so that's why the movement moves towards that inside of the cell to try to equalize, and it's just a natural movement. That's considered passive. In the facilitated diffusion here, again, we have a higher concentration on the outside, moving to the lower on the inside. The only difference is the shape of this molecule is probably what makes it a little difficult for it to just come through on its own. So there is a protein that has a channel for it to pass through. There's no energy being expelled here. It's just a bigger opening to allow for that more awkward shape to come through. That's the facilitated diffusion. So that's why it's also considered passive. And then over here we have our active transport, and in this case we have a low concentration outside. There's high concentration inside, but we're still trying to move these molecules in. And so that's why it's considered active. It's going against that concentration, and to do that we had to use energy here, and that's considered active. Another example of active, of course, are the exocytosis and endocytosis, and these involve vesicles, these little vesicles here. And exo means to go out of, exit, exocytosis, so out of the cell. Endocytosis, endo means to go into, so into the cell, so that's the difference. Now there are some different kinds of endocytosis, or going into the cell. Phagocytosis is cell eating. Pinocytosis is cell drinking. So phagocytosis deals with solids, so things like bacteria. If it's an immune cell trying to bring in a bacteria cell to destroy, that would be considered phagocytosis. Pinocytosis is cell drinking, so bringing in fluids. And then receptor-mediated endocytosis. This is where there's an actual receptor that is going to trigger the vesicle to form and bring in a specific substance into the cell. This might be involved in something like hormones that are going to only interact with specific receptors. So those are the different types of active and passive. And then I wanted to go back to this slide, which deals with the hypertonic, isotonic, and hypotonic solutions and osmosis. So osmosis is another passive transport, and it deals mainly with water, but moves from high to low concentration. And when we talk about that movement of water, we usually refer to substances that are surrounding a cell as either being hypertonic, hypotonic, or isotonic. So in this first diagram here, we have a hypotonic solution. Hypo means less, so the surrounding fluid, the surrounding solution has less dissolved substances. So that means it's got a lot of water in that outside solution. So that would be like sticking the cell in pure distilled water, or sticking it in a very high water content, possibly vinegar or something like that. If you've ever done the egg lab where you stick the egg in vinegar and the shell dissolves and then you leave it there and the egg gets bigger and bigger, and that's because the vinegar is mostly water compared to the inside of the cell. So what happens is, because there is more water outside of the cell and there's less solutes, the hypo refers to the solutes, okay? So there's less dissolved substances, then the water is going to want to flow into the cell to try to equalize and try to dilute the inside of the cell down. And so that's why the cell gets larger. And so I think of hypo, it has the big O in it. So the cells get large and big because the water is coming into the cell. In some cases, it will lyse if it's an animal cell. Now, in plant cells, this is a good thing. We want, that's how they keep their shape. So this would be considered normal for them, and that's how they get nice and strong and taut, and are able to stand upright. The hypertonic, on the other hand, hyper means a lot. So this is where the outside of the cell, the solution, is going to have a lot of dissolved substances. So this would be like a sugar solution, or molasses, or very dense salt solution. Okay, so lots of dissolved substances, very, very little water. And so for this reason, the water inside the cell is going to come out, and that's what causes the cell to shrink, because it's trying to dilute the outside. And so the cell will shrivel up. And you can see in the plant cell this can also happen, and that vacuole's gotten a lot smaller, and that's when you'll start to see plants will start to wilt and wither and fall over. They won't be able to hold their shape. That usually means they need water. They have very little water. Isotonic is what we strive for everything to be in, an isotonic solution. Iso means equal, and that is where the solute concentration on the outside is equal to the solute concentration on the inside. So the water is going to have a net change of nothing. So water coming in and out basically is already at equilibrium, so there's no main changes. And so, and again, that would be okay for a plant cell as well, which it's a little smaller than you would want, but it's still there. All right, any questions on those types of solutions? Okay, all right, so now we're ready to do some practice problems. So starting with this one, this is going to deal with the size of a cell. So we have a simple cuboidal epithelial cell that lines the ducts of certain human exocrine glands. Various materials are transported into or out of the cells by diffusion. The formula for surface area of a cube is 6 times s squared, and the formula for the volume of a cube is s cubed. So s is the length of a side of a cube, and it tells you what the lengths are of each of these. So which of the following cube-shaped cells will be the most efficient in removing the waste by diffusion? So what do we think that would be? Correct. It is definitely A. Definitely going to be A. And yeah, you almost don't even have to do the math here. I mean, it gives you the formulas and the information necessary that you could go ahead and calculate it all out. But ultimately, if we remember that this, from the surface area to volume, the smaller the cube, the better, the higher the surface area to volume ratio is going to be. So it's going to be the most efficient. Okay. So yes, A is correct, because as you start to move up, we're going to start to get a smaller and smaller ratio. We want a large ratio so that we can get more efficiency. So A is correct. Excellent. All right, next question. So here we're looking at endo and exocytosis. We have a pathogenic bacterium that's been engulfed by a phagocytic cell as part of the nonspecific immune response. Which of the following illustrations best represents the response? So in A, we have the bacterium being engulfed, and then it's going over to some ribosomes, and something's happening there, and then getting expelled out. Whereas in B, the bacterium's coming in, and it's going over to antibodies, and then something happens, it's basically destroyed, and then it's going out. In C, it's engulfed, it's taken to lysosomes, where it's broken down and then expelled out. Or in D, it's engulfed, taken to mitochondria, and then expelled out. So which of these is correct based on what we know with cell organelles and what they do? B is a good choice because of the antibodies in there. I see why you would think that. And I was kind of toying with whether I should include this question or not because we didn't really talk about immune responses. But when we talk about antibodies, they are not usually on the inside of the cell. Antibodies are usually markers on the outside of the cell. Okay, and so that B is not going to be correct because it's not being represented correctly here. All right. Normally, yes, if we were talking, so this is kind of not only taking in your information as to which organelles do certain things, but you also do have to know a little bit about the immune responses and how antibodies work. And so B is not representing the correct use of an antibody. So antibodies are not usually on the inside of the cell. They're usually markers on the outside of the cell that would trigger the cell to take in the bacterium. But the actual thing that breaks the bacterium down is going to be the lysosome. So the correct answer here should be C. Okay, so the lysosomes are what do the digestion and the breakdown. Antibodies, yeah, antibodies usually do act as markers, and they are going to be on the outside of the cell, usually a little Y-shaped marker that's on the outside of cells that help to attract certain things to come in. So it kind of marks the bacterium, or it marks the immune cells, so they know what they're supposed to, where they're supposed to be going. Okay. So in this case, the answer would be C, because that ribosomes do not have anything to do with breaking down bacterium. They make proteins, and mitochondria convert to ATP. So again, they wouldn't have anything to do. So as far as what's left, lysosome is the digestive organelle. So that is the correct answer here. All right, this is going to be looking at a graph and osmolarity of a solution. So paramecium are unicellular protists that have contractile vacuoles to remove excess intracellular water. In an experiment investigation, the paramecia was placed in salt solutions of increasing osmolarity. The rate at which the contractile vacuole contracted to pump out excess water was determined and plotted against osmolarity of the solutions as shown in the graph. Which of the following is the correct explanation of the data? So here, if we're looking at the graph, we can see the rate of the contraction is going to, the contractions per minute is very high at the lower osmolarity. As the osmolarity of the solution increases, that rate of contractions is decreasing. So which of these statements would be best to support this data? Okay, A or B? So let's look at those. So with A, it says at higher osmolarity, so the osmolarity of the solution, we know graphs, this would be 0, and so it's going to increase as we go. So at higher osmolarity, we'll say it's like right around this area, the lower rates of contraction are required because more salt diffuses into the paramecium. All right, so we got to remember what we're talking about here with osmolarity and of a solution. So the lower rates of contraction are required because more salt diffuses into the paramecium. So is salt primarily going to be what's diffusing in osmosis or water? Water. So this is saying salts diffusing. So that's going to make this not correct because we're not going to have diffusion of salt. We're going to have diffusion of water. Okay? So then we look at B. The contraction rate increases as the osmolarity decreases. So decreases down here and the contraction rate is up here. So that's correct so far. We do have, at the lower osmolarity, we do have an increase in the contractions because the amount of water entering the paramecium by osmosis increases. So it is the movement of water and it's entering. That is correct. Okay, so B is the correct answer because the osmolarity of the solution is lower. So that means that the salt is going to be lower. So there's going to be more water content in that solution. So that's going to force the water into the paramecium. So this says the amount of water entering is going to increase. And that's going to cause that paramecium to have to get rid of the water because otherwise it might explode. So does that make sense? Okay. All right. So lower osmolarity, the lower osmolarity of the solution means that it's going to have less salt and more water. So that means the water is going to go in. And because it's going in so much, the contractions have to increase to get it out because otherwise the paramecium will explode. Now C, just to kind of go through them, this talks about the contractile vacuole is less efficient in solutions of high osmolarity because of reduced amount of ATP. Again, the ATP is not going to be necessary because it's osmosis, so we don't need to worry about that. And cellular respiration doesn't have a role here in this particular, with osmolarity and movement there. So it has nothing to do with that. So that's not right. And then D, in an isosomatic salt solution, there is no diffusion of water into or out of the paramecium, so the contraction rate is 0. Well, we're not, that's not something we can verify from this particular graph. We don't know where there might be an isometric existence because it never intersects at either point. So we don't have enough information for that particular one. Okay? All right, next question. Both myoglobin and hemoglobin are proteins that bind reversibly with molecular oxygen. So they both bind with oxygen. The graph below shows the oxygen binding saturation of each protein at different concentrations of oxygen. So what we want to see is basically we're looking at this graph and trying to see which of these following statements is correct. So A, at 10 millimeters of mercury pressure, so that's 10 is right here. Hemoglobin binds oxygen, but myoglobin does not. Is that a correct statement? Correct. Good. No, it is not a correct statement because the y-axis is showing us saturation with oxygen, so the percent of oxygen on that particular protein. And at 10, we definitely have some, because of our intersection here, we have what, 50, 60, 70, 80, it's probably about 80% saturated. So that's not correct. Myoglobin does have oxygen. So B, at 20 millimeters, so that's here, of mercury, at 20 millimeters pressure, myoglobin and hemoglobin bind in equal amounts. So this is the hemoglobin intersection and this is the myoglobin. Does that equal amounts of oxygen? No, it's not. And you are correct. You've already put down that C is the correct answer, and that is correct. So at 40, which is right here, myoglobin has a greater affinity for oxygen than the hemoglobin does. And if we draw that up, we can see that there is a greater affinity for the myoglobin at that point. D is not correct either. It says at 80 millimeters, which is over here. myoglobin binds twice as much oxygen as hemoglobin, and that's obviously not correct because they're almost at the same levels at this point. All right, excellent job. Okay, now this is a free response question, so just kind of wanted to give you an idea of what a free response is. This is one of the ones that would only be worth 3 points, and just to kind of give you an idea of what some of these kind of look like. So this says the following data were collected by observing subcellular structures of 3 different types of eukaryotic cells. So we have 3 different cells, X, Y, and Z, and this gives us data on what types of organelles are in those different cells. And so the question's asking you to, based on the analysis of the data, identify a likely primary function for each cell type and explain how that data supports the identification. So you're going to be asked for each of these to give a primary function and then explain using the data why you think that is the case. And so it'll be 1 point for each of the cell organelles. So for cell X, it's got a small amount of smooth ER, a small amount of rough ER, a large number of mitochondria, does have some cilia present, and a small amount of Golgi bodies. So what might be a possible function for X? And you would want to write in complete sentences, but I'm not, just to save us time here. Have any ideas or suggestions? Okay, an energy generator as its purpose. That's not really a function of a cell necessarily, is just to produce energy, but what kind of cell would need a lot of energy? What would it be doing? What kind of function would require a lot of energy? A blood cell. Well, blood cell's movement is moving oxygen, and it flows through the liquid, so it doesn't usually have to move itself. So I'm not so sure that it would need a lot of mitochondria, a lot of energy. So I would actually say probably it's going to be the function would be some kind of movement. That would be my best guess. Primarily because of the large number of mitochondria. Yeah, muscle cell. Now, muscle cell would not be the good answer here because that's giving the name of, that's not really giving the function. But if you could think that, okay, this would maybe be a muscle cell, what do muscles do? They are used for movement. So the function would be movement, and an explanation to back that up would be because of the large number of mitochondria. And I'm just going to draw an arrow there, okay, that it says it has. Now, what about Y? Y has a large amount of smooth ER, a large amount of the rough ER, a moderate number of mitochondria, no cilia, but a very large, or a large amount of Golgi bodies. So what might be something that you might see this cell doing? This one I found would be a little bit more difficult to think about because it's kind of vague. Function could either be moving things in an analysis cell or possibly storage. Okay, those are good options. I would, definitely agree with you on the possible storage or movement of stuff in and out because of the Golgi body and the ERs. And I'll put because of that. And again, you would want to be writing all this in complete sentences and make sure you give your details to back it up. Now Z is interesting because it says that it has no smooth ER, rough ER, mitochondria, cilia, or Golgi apparatus. So C doesn't have any of those things. So what do you think Z might be involved in? Right, could be a dead cell. That's actually a very good answer. Could be a dead cell. And I would say to back that up is because there are no functional organelles at all, okay? So let's just take a look at what the grading that it goes with this, what the readers would be looking for as correct answers here. So for X, it said that one of the functions could be locomotion, or you might have said movement or surface transport. And then the explanation to support that would be it has cilia for movement and a large number of mitochondria, or movement of particles along the cell surface because of the cilia. Okay? So that's where the identification, the backup would be. For Y, the two main choices it gives here are the secretion or exocytosis. So you mentioned the movement of things in and out, so that would fall under that exocytosis. And you would need to back that up with having large amounts of the ER and Golgi apparatus to package those proteins, or protein synthesis would have been acceptable as well. Another option for Y that they would accept would be like lipid or hormone synthesis, or detoxification, and the backup for that is because of the large amounts of smooth ER, because the smooth ER deals with lipid synthesis. And so that would be your explanation. The ER, there's large amounts of ER that helps to produce lipids. Z, we had put it as a dead cell, which I think falls under no function. So you would, it has no function, it's dead, and that's because it is does not require any of these organelles. It does not have any of those organelles. Other options that they actually accept here, if you had put transport, it would take that if you backed it up with oxygen transport in animal cells, and that does make sense because, like I mentioned before, the red blood cells kind of eject out their nucleus and other organelles to make room for oxygen as they mature, so it's possible that it would not have hardly any other organelles in it. Protection was also an acceptable answer here with the backup that epidermal cells, so like your cells on your skin, the stratum corneum, or your nails, those cells tend to, as they age, they have less and less organelles in them because they're basically becoming dead, so they do not require any of those organelles. Support is also an acceptable answer here if you reference ground tissue or vascular tissue in plants, because it does not require, again, these organelles. And then storage was also acceptable if you talk about how not having those organelles maximizes the volume and space that's available inside the cell. So just some examples of some, there are definitely different ways to look at how things can be answered. The key here is you can see that the only way to get these points is if you not only identify the function, but explain how the data supports that identification. It says no points for identification without explanation. So it's very important that you don't forget that step. So that's one of the things I wanted to kind of show you in how that free response questions go, is that there has to be a complete follow through. Okay? Any questions? All right. Well, Caleb, that is it for today. Thank you for joining me, and I hope you have a wonderful afternoon. And I'll see you again on Thursday, hopefully. Okay? Thank you. You too.